Restriction fragment length polymorphism. Restriction fragment length polymorphism is a technique in which organisms may be differentiated by analysis of patterns derived from the cleavage of their DNA. It is a technique that exploits variations in homologous DNA sequences. Here, I mean homologous is the same type of DNA sequences. A restriction fragment length polymorphism is defined by the existence of alternative alleles. Allele, it is an alternative form of gene associated with restriction fragments that differ in size from each other. Simply, the variations in the restriction DNA fragment length between an individuals of a species is called RFLP. RFLP, none other than restriction fragment length polymorphism. The basic technique of identifying such restriction fragment length polymorphisms involve fragmenting a sample of DNA by a restriction enzyme. Restriction enzyme is an enzyme which involves in the cutting of the DNA molecule, which recognize and cut the DNA wherever a short sequence occurs in a process known as restriction digest. Then, the resulting DNA fragments are separated by length through a process known as agarose gel electrophoresis. It is then transferred to a membrane via Southern blot procedure, followed by hybridization of the membrane to label DNA probe, then determines the length of the fragments which are complementary to the probe. And RFLP occurs when the length of a detected fragment is between individuals. The picture clearly depicts restriction fragment length polymorphism. Here, the blood stain is taken. From the blood stain, the DNA is extracted and it is then subjected with restriction enzyme. The restriction enzyme further cleaves the DNA molecules. So, as the restriction enzyme cleaves the DNA molecules, the fragments of DNA with varied length are produced, and these fragments are further separated by electrophoresis. The fragments are then transferred to a membrane through a technique called Southern blotting, which has been discovered by E.M. Southern, and it is then transferred into a radioactive DNA probe, where the DNA probe binds to specific DNA fragments and the membrane is washed free of endless probe, and it is then detected through X-ray film and sandwiched to the membrane to detect radioactive pattern. And finally, the DNA pattern is compared with patterns from unknown subjects. Here comes the objectives of RFLP. In paternity cases or criminal cases, to determine the source of a DNA sample, that is, it is used in forensic applications, and in the detection of mutation, to determine the disease status of an individual, and to measure the recombination rates which can lead to a genetic map with the distance between RFLP loci. In the characterization of genetic diversity or breeding patterns in animal populations, RFLP has been developed for chromosome mapping of humans, mice, maize, tomato, rice, etc. Thank you.